When I first saw the Krebs cycle and everything we needed to memorize, the intricacies that go into the cycle, my mind was overwhelmed. Today, I'm going to help you break down the Krebs cycle into simple steps and give you tricks to understand all of the details. This biochemical pathway is an amazing part of our cells' activities to give us energy, so even with all there is to know, it is worth the effort. To begin, the Krebs cycle, also known as the citric acid cycle, is used to make energy through oxidation of acetyl-CoA. Cells consume oxygen, and from the cycle, CO2 is produced. The three main stages are acetyl-CoA production, acetyl-CoA oxidation, and electron transfer and oxidative phosphorylation. We're going to focus on the first two steps, which happen during the citric acid cycle. The first step happens to prepare for the cycle. Before the cycle begins, glucose needs to become pyruvate. It starts as a 6-carbon molecule and through glycolysis is broken down to 2 3-carbon molecules of pyruvate. Then this 3-carbon pyruvate is oxidized to acetyl-CoA by the enzyme pyruvate dehydrogenase complex. In this reaction, a molecule of CO2 and a molecule of NADH is created. Acetyl-CoA is a 2-carbon compound. Next, the acetyl-CoA joins oxaloacetate, which is a 4-carbon compound, and citrate is the resulting 6-carbon compound. Since citrate is synthesized, the enzyme that helps the reaction is citrate synthase. This step limits the rate of the cycle. It depends on the amount of oxaloacetate, which is kept low to regulate the cycle if there is a high amount of NADH and the cell does not need more. A water molecule is lost and becomes COSH, a coenzyme that functions to accept and carry acetyl groups. The citrate is then isomerized to isocitrate with the enzyme aconitase. Since citrate is a tertiary alcohol, it is not fit for oxidation. Therefore, to be useful, it needs to be dehydrated and then rehydrated, causing it to become its isomer isocitrate, which is a good substrate for oxidation. In its ideal secondary alcohol form, isocitrate can be oxidized to alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. At this point, I would like to introduce an acronym I learned to remind me of exactly what each reaction does. With the acronym OIL-RIG, we can remember that oxidation is losing an electron and reduction is gaining an electron. So when isocitrate is oxidized to alpha-ketoglutarate, the electrons are lost, but are gained when NAD is reduced to NADH. Oxidation is losing, reduction is gaining. This is the first reaction in this cycle that shows oxidation-reduction coupling. As we go back to the cycle, we see that the resulting alpha-ketoglutarate from the oxidation of isocitrate is a 5-carbon molecule, and since it came from the 6-carbon compound of isocitrate, in this reaction, 1-carbon molecule is lost and forms a carbon dioxide molecule. Alpha-ketoglutarate is converted into succinyl-CoA with the enzyme alpha-ketoglutarate dehydrogenase. This is the last oxidative decarboxylation, just like in the previous step of the cycle. Again, the oxidation of alpha-ketoglutarate to succinyl-CoA is coupled with a reduction of NAD to NADH. Electrons are lost, then gained, and through the decarboxylation, another carbon molecule is released in the form of a carbon dioxide molecule. Succinyl-CoA is a 4-carbon compound after the release of CO2 and is a high-energy thioester bond, which we will need for the next reaction. A thing to know about these two oxidative decarboxylations, where NADH and CO2 are formed and released, is that they are highly thermodynamically favorable and irreversible, meaning that they have a negative delta G energy, and that once the process has the correct product input, it happens without the need of energy and stabilizes by creating a high to low energy gradient. That also means that these steps can be regulated with product inhibition, so if the previous product doesn't go in the process, it does not continue. Succinate is then synthesized from succinyl-CoA with succinyl-CoA synthase, and here a molecule of GTP is created. The previous high-energy thioester bond from succinyl-CoA allowed for a phosphate group to be incorporated, and when the phospho group is transferred in phosphorylation, GTP is produced. This step is reversible, but the product input concentration is kept low to pull the reaction forward, making it slightly favorable. Our last 3 steps of the cycle are mainly for replenishing oxaloacetate. These anaplerotic reactions are the way we can keep the cellular respiration going. Succinate is converted into fumarate by succinate dehydrogenase. This flavoprotein is attached to the mitochondrial inner membrane and is part of complex 2 in the electron transport chain. In this reaction, FADH2 is created and the product concentration is kept low to pull it forward because the thermodynamic energy is near to equilibrium. Fumarate is then converted into malate by the enzyme fumarase through hydration across a double bond. In the last step, malate is converted to oxaloacetate with malate dehydrogenase. And as we have seen previously in the cycle, when you see the word dehydrogenase, an oxidation-reduction coupling is going to happen. NAD+ is reduced, and our final NADH is produced. This reaction is highly thermodynamically unfavorable because energy and product concentration is kept extremely low by citrate synthase, so that the reaction is pulled forward. It could not happen spontaneously like the favorable reactions, and has a mutual relationship with acetyl-CoA, as they need each other to start the citric acid cycle again. As we look at all of the products of these steps, we can summarize that through one cycle, 3 NADH, 1 FADH, 1 GTP, and 2 CO2s are all formed. And since 1 glucose was changed to 2 pyruvate molecules to fuel the cycle, all processes happen twice for each glucose. So, from 1 glucose, we receive 6 NADH, 2 FADH, 2 GTP, and 4 CO2. From glycolysis of pyruvate, we receive acetyl-CoA to start the cycle, and by the end of the cycle, oxaloacetate is used and later regenerated. After quite possibly boggling your brain with all these processes and net products, I will leave you with some fun facts of the citric acid cycle so you can now understand how the cycle as a whole works. The citric acid cycle is an amphibolic pathway. This means that it has both catabolic pathways for breaking down larger molecules leading in, and anabolic pathways for replenishing the cycle by building up molecules leading out. Intermediates in this cycle do not just serve the cycle, they can also be used independently as biosynthetic pathways. When highly thermodynamically favorable and irreversible steps or exergonic reactions happen, they are highly regulated, meaning that the cycle does not continue until each one goes smoothly. These steps are the enzymes pyruvate dehydrogenase complex citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. The cycle is activated when substrate is available to start the cycle, or when there is a low amount of NADH, NAD+, or ATP, ADP. It is inhibited when products accumulate, and there is no need for the cycle to make more, like when there is a high amount of NADH, or NAD+, ATP, or ADP. Feel free to go back and go over any part of this video that confused you. And remember, you can learn anything.